So related to diffusion is osmosis, which specifically describes the diffusion of water molecules. Fortunately, it follows all the same rules that I've just talked about with just one complication, which is that we never find pure water anywhere inside the body. It always has solutes dissolved within it. Now, you might remember from some of your other courses that mass displaces volume, so Archimedes in the house. But if you take a look at the top image on this slide, you're seeing a compartment made up of pure water. It has 25 blue water molecules present within it. Just below that, you're seeing a container with an equivalent volume, but it has 5 molecules of solute dissolved in the water. And that means there's only room for 20 molecules of water in that compartment. So even though the two containers have equal volumes, they actually have different concentrations of water. Now, quite honestly, there are a lot of different ways that people explain osmosis, but I think the easiest thing to remember is that solutes lower the concentration of water. So that means that a compartment that has a higher concentration of solute effectively has a lower concentration of water. And that's important because water, just like everything else, can move by diffusion from relatively higher concentration to relatively lower concentration. So in this selectively permeable tube that you're seeing on the right-hand side here, the water is going to move from the left side of the tube into the right side of the tube. And that causes the left side to decrease in volume and the right side to increase in volume. Here's a couple more examples. On the left side here, you're looking at two compartments that are separated by a membrane that's freely permeable to both water and to solutes. So in the top image, we start off with different concentrations of solute. There's 2 in the left side and 4 in the right side. And we have different concentrations of water molecules too. Now, because the membrane's freely permeable to both water and solute, they're both able to move down their concentration gradient until the concentration gradient no longer exists. So if you look at the bottom panel, you'll see everything is completely balanced. Equal concentrations of solute, equal concentrations of water on both sides. The net flux is 0, and the containers have equal volume. On the right side, we have the same initial conditions, but we have a selectively permeable membrane. It's permeable to water, but not to solute. So in other words, the water is free to move, but the solute isn't. So if you take a look here, you'll see that the solute concentration, again, in the right side is higher. It's 4 versus 2 on the left side. But remember, this means that the concentration of water on the right side is lower. And you can actually see that if you count the number of water molecules. Because there's a concentration gradient for water, it's going to move from the area of higher concentration, which is the left side, into the area of lower concentration, which is the right side. And that means the left chamber is going to lose volume, the right chamber is going to gain volume, and all that's because of the water. The solute didn't move at all. I guess the major takeaway from this quick discussion of osmosis is that the movement of water is often linked to changes in cell volume, and that happens when solutes are unevenly distributed between two compartments. We don't typically want our cells to change their volume, so we have to try to regulate the solute concentrations very carefully in order to limit the amount of osmosis that occurs. So when the concentration of solute falls out of homeostasis, we can apply our knowledge of osmosis to make predictions about how this would impact the cell. The relationship between volume and solute concentration is known as tonicity. And just like the anatomical terms, it's a relative term that always requires a comparison between two compartments. Now, by convention, when biologists talk about tonicity, they're describing the concentration of the outside environment relative to the inside environment. So in the event that you have an increase in solute concentration outside the cell, that solution becomes hypertonic. Remember that higher solute concentration means lower water concentration. So this is going to cause water to move out of the cell into the extracellular fluid and lose volume. In contrast, if we reduce the solute concentration in the extracellular fluid, we create a hypotonic solution. When we have fewer solutes outside of the cell, that means that there is a higher concentration of solutes inside the cell, so there's a lower concentration of water. So in that situation, water comes in, the cell increases in volume, it swells, and usually actually loses its structural integrity, because remember, the plasma membrane doesn't provide any physical protection. Now, we don't want our cells to be shrinking or swelling. We normally want them to maintain their normal size. And that means that the solute concentrations have to be balanced. We want the extracellular concentration of solutes to be equal to the intracellular concentration of solutes. In other words, we want them to be isotonic. In the event that we have a mismatch and we are experiencing some of those osmotic forces, the pressure that's created by the water, it often has to be balanced out by some kind of counterbalancing force. Now, usually this is known as a hydrostatic force. It's the force that's created by a fluid kept under pressure. And we're going to talk more about this counterbalancing force later when we get to the cardiovascular system. Another special case with diffusion is known as facilitated diffusion. Now again, it follows all the same rules that we've talked about up to this point. The only difference is that the solute in facilitated diffusion requires the help of an integral membrane protein in order to be soluble. An example of this that we'll see many times in this course is with glucose transporters. They help to bring glucose into your cells following a meal when the glucose concentration is high inside your bloodstream. But because glucose is so large, it's not able to pass through the membrane, and so it requires the help of a carrier protein. Probably the weirdest case is bulk flow. Again, bulk flow is a passive transport process, but it's weird because it can break some of the rules that we've established for simple diffusion. It's the only passive transport process that can move molecules independently of their concentrations. Now, in order for bulk flow to happen, you don't need a concentration gradient, but you do need a pressure gradient. And as the pressure equalizes, we move from high pressure to low pressure, the solutes are carried along with it. This is similar to what might happen if you were to throw a rock into a moving river or stream and it just got swept away in the current. Or in my case, because I'm a science fiction nerd, this is essentially what happens when someone gets thrown out the airlock. A spaceship is under relatively high pressure. Space is very, very low pressure. So when someone opens a door, that pressure equalizes. Everything moves out into space, and the solutes, or in this case, the people, get sucked out there with it. This example was taken from Battlestar Galactica, but we've also seen this in two of my other favorite sci-fi shows, which are Star Trek and The Expanse.